So you have seen that how we form the uracil based pyrimidine nucleosides. Now let's move towards the other type of nucleosides, specifically, for instance, the cytosine based or CTP production. This is once you have formed the UMP, which eventually can be very easily be converted into the UTP. This UTP then can be aminated to form the CTP, and you can see here again the amine group is provided by the glutamine, which is converted into glutamate, and of course this is again an energy intensive reaction, and you will ultimately get the CTP or cytosine triphosphate. As was case with the UTP, similarly CDP, that is the diphosphate, that is the cytosine nucleoside consisting of 2-phosphate moieties. The CDP can also be then converted into their corresponding deoxyribonucleic acids through the activity of the ribonucleotide reductase. Once you have the what we say, you got this deoxy CDP, it can be converted to deoxy CMP, and this deoxy CMP then give rise to the formation of deoxy UMP, which then can be converted into deoxy TMP, that is thymidine monophosphate, and this thymidine monophosphate then can be converted, deoxythymidine monophosphate can be converted into deoxythymidine triphosphate, which will eventually become a part of the DNA. The enzymes here are thymidine synthetase, and also there is also what we say the another cofactor which is involved here is the methylene tetrahydrofolate, which is reduced to what we say the dihydrofolate. And so here again, as we have been seeing, that you can target these enzymes, for instance here thymidine synthetase, by different inhibitors, which can then inhibit the formation of for instance the DNA molecules. For instance, if there is the inhibition of thymidine nucleosides or deoxythymidine nucleosides, what will happen? It will eventually lead to the inhibition of the formation of DNA, and then it can be a target for the cancer cells, because we have already told you that cancer cells they need a continuous replication of their DNA so that they can divide and they can then proliferate. So in this way, you have seen that when we are making different moieties, different purine or pyrimidine molecules, we also can change these nucleosides into their corresponding deoxynucleosides, which can be, or deoxynucleotides, which can be used in the formation of DNA. So there is always a balance between the formation of nucleotides and deoxynucleotides depending upon the need of the cells. Neither of these can be compromised. Sometimes you need, of course, that's a major need of your cell is to have DNA, but you know that if you have excessive formation of DNA, that is what cancer cells do. Similarly, you also need RNA, because we know that RNA, in addition to a messenger RNA, which is involved in the formation of proteins, you also need other RNAs. And now we know that there are many regulatory RNAs which are very indispensable for our survival. So that's why there is always a balance between the formation of nucleosides and formation of deoxynucleotides.